Pharmapal.
Proteins, ligand binding stability, molecular dynamics, and cross-docking analysis. Welcome back to our channel. In today's video, we're diving into the fascinating world of target selection and drug discovery, specifically focusing on the recent study investigating the stability of ligand binding modes to proteins through molecular dynamics simulations. What is docking? Before we get into the details, let's clarify what docking means. In the context of drug discovery, docking refers to the computational process of predicting how a small molecule, or ligand, interacts with a protein. The goal is to find the best binding pose, which is crucial for designing effective drugs. The challenge of docking. One of the main challenges in docking is selecting the correct pose from many potential candidates. Researchers use various scoring functions to evaluate these poses, but distinguishing the right one from decoys can be tricky. This is where this study comes into play. Key objectives of the study: investigate the stability of ligand binding poses using molecular dynamics (MD) simulations. Compare different docking protocols to see how they perform in practical scenarios. Could you assess the effectiveness of MD simulations as a filtering tool for docking poses? Methodology overview. The researchers selected 3 target proteins of pharmaceutical interest. Thrombin, heat shock protein 90-alpha, HSP90A, cyclin-dependent kinase 2, CDK2. They used various docking methods, including normal docking, induced fit docking, IFD with homology models. For each ligand, they generated 3 poses. Pose 0, the closest to the crystallographic structure, the correct pose. Pose 1, a high-ranking pose but different from pose 0. Pose 2, another high-ranking pose, also distinct from pose 0 and pose 1. Findings from molecular dynamics simulations. The study revealed that correct poses, pose 0, were generally more stable than decoy poses, pose 1 and pose 2, during MD simulations. The ability to discriminate between correct and decoy poses depended on the criteria used for stability assessment. For example, if at least one of 5 MD runs showed stability, 95% of correct poses were retained. If 2 or 3 runs were required for stability, 31 to 56% of decoys could be excluded, but there was a risk of losing some correct poses. Next, we'll break down the key figures and concepts discussed in the paper, making it easy to understand even if you're new to this field. So, let's get started. First off, let's talk about target selection. In previous research, they compiled a dataset of 1,039 protein databank (PDB) files consisting of drug-like ligands. From this extensive dataset, they selected 3 protein targets that are of significant pharmaceutical interest. Thrombin, heat shock protein 90-alpha, HSP90A, cyclin-dependent kinase 2, CDK2. These proteins are not just interesting, they are already targets for marketed drugs, making them ideal candidates for our current study. Complex structures for cross-docking. For this cross-docking studies, they chose a total of 27 complex structures for thrombin, 41 for HSP90A, 50 for CDK2. This selection was crucial as it provided a solid foundation for our systematic study. Now, let's move on to how they generated the initial poses for our simulations. They employed various docking protocols using the Schrödinger software. For thrombin, they selected 2 templates, PDB codes 3DA9 and 3QX5, for docking. For HSP90A, they utilized the induced fit docking, IFD, method to account for known ligand-induced structural changes. For CDK2, they performed homology modeling based on the CDK1 template, PDB code 4Y72. This variety of approaches reflects the complexities of real-world docking situations. Pose definitions. T. Hay defined 3 poses for each ligand. Pose 0, closest to the crystal structure, RMSD less than 2.0A. Pose 1, smallest G-score but deviates from pose 0. Pose 2, another small G-score pose, distinct from pose 0 and pose 1. This classification helps distinguish between correct poses and potential decoys. Molecular Dynamics Simulation Protocol. Next, let's briefly touch on our Molecular Dynamics (MD) simulation protocol. They performed 2 rounds of docking to generate comparable poses. They used a simple docking method for thrombin, while for HSP90A, they combined it with IFD. For CDK2, they relied on the homology model and IFD to predict binding modes. RMSD calculations. To evaluate the stability of these poses, they calculated the root mean square deviation, RMSD, values, which helped us understand how closely the poses maintained their structure during simulations. Pose 0 generally showed more stability compared to the decoys, indicating its reliability as the correct pose. In our results, they found that the correct poses are more stable than decoy poses during MD simulations. The discrimination capability of MD simulations varied based on the criteria used for stability assessment. This means that if they judged a pose as stable based on just one of 5 parallel MD runs, they could retain about 95% of the correct poses while excluding a significant number of decoys. In conclusion, the study suggests that simple equilibrium MD simulations can effectively filter out decoy poses that docking scores cannot distinguish. This approach can save time and computational resources in drug discovery. If you found this summary helpful, please give us a thumbs up and subscribe for more insights into molecular modeling and drug discovery. Let us know in the comments if you have any questions or topics you'd like us to cover next.
Pharmapal.